Thanks, Znola, for the invitation to talk about immunotherapy in the treatment of high-grade glioma, particularly in the treatment of glioblastoma multiform. The underlying mechanism involved in the pathogenesis of glioblastoma multiforme is presently unclear, but we do know that the immune system has a key role in the progression and the development of this kind of tumor. We can see many mechanisms of stimulation of lymphocytes that are suppressive in this tumor. And alongside this phenomenon, we can see that inhibitor mechanisms of the immune system, and particularly T cells, they are upregulated in this tumor. This understanding is very important to the research that are ongoing nowadays in this tumor, and recognize this phenomenon was important to develop many vaccines to boost the immune system against this kind of tumor, and how these vaccines are being doing today. The first of all of these vaccines is the more advanced stage is the vaccine against EGFR V3. There is a protein present in around 30% of the glioblastoma multiforme and is lacking in the normal tissue, brain tissue. And this vaccine it's intradermal vaccine and can elicit the immune system against the EGFR V3 of glioblastoma multiforme that harbor this protein. The result of phase 2 trial were very promising, and we could see 26 months of overall survival, huge increase in the overall survival comparing to the overall survival seen in the STUP trial that was 14%. We participated in 2014 there in Beneficência Portuguesa in São Paulo of the phase 3 trial of rindopepimode. That is the name of the vaccine against EGFR-V3. We are waiting in the forthcoming month the results, the definitive results, but we have the hope that this vaccine could be good for the patients. The next vaccine there is harnessing the immune system against glioblastoma. It's vaccine with heat shock protein. In the last ASCO in June in Chicago, we could in 2015 we could see an impressive result from Dr. Oren Block from Southwestern University in Chicago that showed a heat shock protein produced a 44-month overall survival in patients with glioblastoma multiforme that expressed in the circulating monocytes PD-L1. It was absolute amazing results. We could not see such results in the last trials, and we cannot forget that we're talking about GBM, a devastating disease with very few overall survival, but this result could bring us many concern and many attention for the development of this kind of vaccines. Let's wait, and I think that ASCO 2016 we could see the follow-up of this trial. The next vaccine that is being that has been developed in the treatment of glioblastoma multiforme is vaccine with dendritic cells. We know that dendritic cells are the major antigen-presented cells in our body, and antigen presented in the surface of glioblastoma multiforme are presented to dendritic cells in the lab, and this dendritic cell has been given to the patients through a vaccine, and can elicit again immune response against glioblastoma multiforme. We recognize that around 90% of the cytomegalovirus genome as genetic material can be detected inside GBM, and this is an important clue to the development of vaccines with cytomegalovirus antigens, and this is an ongoing field. We must await the result as well. And the last vaccine that I would like to talk to you is with poliovirus. Poliovirus is a DNA virus that has a short-lasting life, and inside the lab, the researcher inactivated a portion of the genetic material of this virus, and used this to put inside the surgical cavity of patients with GBM that was resected, and we can see that the results are impressive, are amazing. 3 years ago, the very first patient was enrolled to this trial, and this patient is still alive and without evidence of disease. This is a very promising study. We must wait the next results. And it's hard to miss to tell you the huge role of immunotherapy it's another field in the immunotherapy of all kinds of tumors. It's not different in GBM. In ASCO 2014, we saw 2 papers showing that glioblastoma multiforme has a huge expression of PD-L1, and this understanding is very important to the development of trials using nivolumab and pembrolizumab against this tumor. Another molecule, inhibitor molecule, that is being exploited and explored in the treatment of glioblastoma is CTLA-4. This is an inhibitor molecule, checkpoint molecule, and because of these 2 checkpoint inhibitors, PD-1, PD-L1, and CTLA-4, nivolumab, pembrolizumab, and ipilimumab are being used in the treatment of this kind of tumors. Now, it has been already finished a phase 3 trial with 372 patients with glioblastoma multiforme after failure to temozolomide, and this trial has already closed recently, and the results are eagerly awaited. And this trial consists of, after failure to temozolomide, the research gave to the patient or nivo plus ipi or bevacizumab. We must await the results, and I think that immunotherapy came to stay, definitely, and presumably in the next years we could see a revolution in the treatment of this kind of tumors. Thank you, Snow, again, and see you in the next.